In a groundbreaking study led by Penn State, researchers have uncovered the long-term effects of early life exposure to persistent organic pollutants, or POPs, on the gut microbiome. This study, published in the journal Environmental Health Perspectives, highlights how these forever chemicals can lead to metabolic disorders in adulthood. By examining mice, the researchers have provided insights that could explain the rising epidemic of metabolic diseases such as obesity and type 2 diabetes in humans. The study's findings underscore the importance of understanding environmental impacts on health from an early age. The study focused on a specific POP known as 2,3,7,8-tetrachlorodibenzofuran, or TCDF. TCDF is a byproduct of industrial processes like waste incineration and fossil fuel combustion. It accumulates in the food chain, primarily affecting humans through the consumption of high-fat foods such as meat, dairy, and certain fish. Babies can also be exposed through breast milk. POPs are pervasive in the environment, and nearly every living organism has been exposed, said Andrew Patterson, a professor at Penn State. The negative health effects of these chemicals are well documented, including birth defects and cancer. The research team conducted experiments on 2 groups of mice, one exposed to TCDF and a control group with no exposure. The mice were given pills containing 0.46 micrograms of TCDF for 5 days. Although this dose is higher than typical human exposure, it was not toxic. The researchers then assessed the mice's gut microbiomes and health indicators, such as body weight and glucose tolerance. They found that early life exposure to TCDF permanently disrupted the gut microbiomes of the treated mice. These mice exhibited higher body weight and glucose intolerance at 4 months, equivalent to young adulthood in humans. The study revealed that TCDF exposure led to a decrease in certain beneficial bacterial species, including Akkermansia mucinifila, which is crucial for gut health. We found that early life exposure to TCDF permanently disrupted the gut microbiomes of the wild-type mice, said Yuan Tian, lead author of the study. The researchers further demonstrated that mice receiving microbiome transplants from TCDF-exposed mice developed metabolic disorders. This finding indicates that the altered microbiome is a significant factor in the development of metabolic diseases. The study highlights the critical role of the gut microbiome in mediating health outcomes. The implications of this study for human health are profound. The research suggests that early exposure to TCDF and similar chemicals could be contributing to the prevalence of metabolic disorders in adults. The disturbances in the gut microbiome caused by these chemicals may persist long after the chemicals have been eliminated from the body. This raises concerns about the long-term health effects of environmental pollutants and underscores the need for strategies to mitigate their impact. Understanding the relationship between chemical exposure and gut health could lead to new approaches in preventing and treating metabolic diseases. Looking ahead, the researchers are exploring ways to restore the gut microbiome to its optimal state. One promising approach involves using probiotics, such as Akkermansia mucinifila, to counteract the effects of TCDF exposure. Our findings suggest that these bacteria are influenced by toxic exposure and play an important role in mediating health outcomes, Patterson explained. With further research, it may be possible to develop probiotic or prebiotic treatments to restore gut health in individuals affected by environmental pollutants. This study opens new avenues for research and highlights the potential for innovative solutions to combat the health impacts of forever chemicals.